Assalamualaikum everyone. In today's video, we are going to discuss how we count RBCs in the physiology experiment. So, before we are going to start this lecture, I request you to please subscribe to my channel, like my videos, and please press the bell icon to get the notification of all the videos which I have uploaded. So let's start the lecture. So in this lecture we are going to discuss how we count RBCs number which are present in our body. And first, it is very essential for us to count the number of RBCs which are present in us because a lot of disease can be diagnosed with the help of it. Okay, so therefore it is very important. So what we see at the introduction of this, so RBCs make 45% of your blood volume, and its primary function is to carry oxygen from your lungs to your cells. RBCs are composed of protein, some iron compound which is called as hemoglobin. Hemoglobin has a heme group which is iron group, and it has capacity to carry the oxygen and to transport this oxygen to the every cell of your body. Hemoglobin releases oxygen to the cells. RBCs lack nucleus as it packed with the hemoglobin. Okay, so the mature RBC have no nucleus in them because they have hemoglobin in it. Each RBC is biconcave. This is the shape of RBC and having the diameter of 7.2 micrometer and thickness is 2.2 micrometer. So this is the dimension of your RBCs. Okay, and it also contains carbonic anhydrase to carry CO2. So carbonic anhydrase is an enzyme which is helpful in the formation of bicarbonates. Okay, so they are this enzyme helpful for reacting carbon dioxide and convert it into the bicarbonate. Now at the end, when we transfer the carbon dioxide to the desired place, then the carbonic anhydrase again reverse this reaction and make up carbon dioxide again. Okay, so this was the small introduction of RBCs. First, you may say that how percentage of RBCs is composed of your blood volume, what is their function, and what is their role, what is their dimensions, and then now we discuss the aim of this experiment. So the aim of this experiment is to count RBCs and is to determine whether the adequate number of RBCs is present in your body or not. So the method for calculating it is the manual method and the electronic cell counting. So the manual RBC count is given here, an anticoagulant whole using EDTA or heparin. So these are the anticoagulants used as anticoagulants, or blood capillary can be used. So first of all, in this manual method, you have to take up anticoagulant. Okay. Now the Hyman solution, this is the diluting fluid, and the composition of this solution is given here. It composed of mercury chloride and 0.05 gram of it, Na2SO4 2.5 gram, NaCl 0.5 gram, and distilled water 100 ml. So this is the composition of this Heim solution. Now RBC pipette, which is composed of a stem and a mixing chamber with a red bead in it. I have shown to you the RBC pipette in the previous lecture, and its function is to mix the blood with the substance and differentiate from the RBCs, and it's helpful in the detection in the counting of RBCs. So now after adding our blood into this RBC pipette, now we move on to the Neubaer chamber where we are going to count our RBCs. So it is counting chambers with a cover shape. So in the cover shape we have a lot of counting chambers. The same counting chamber are also used for the counting of WBC, but the difference between both of them is that for RBC we are going to use this RBC pipette, and for the WBC there is another pipette which is called as WBC pipette, and the WBC pipette has white bead in it. So the things you need for this experiment is a microscope, you need a lancet to pick out your blood, you need alcohol, and you need cotton. So now let's discuss the procedure of your experiment. So first of all, you have to wipe your partner's finger and with the help of cotton and apply spirit swab on it. Then after it, you have to prick it after drying it. Okay. So when a drop of a reasonable size is collected, you have to ignore the first drop of your blood which is coming out, and then you have to find out the blood with a reasonable size, and after it you have to add up the clots, the drops of your blood into the RBC pipette by placing the RBC pipette slightly tilted in the vertical position to your finger. Now apply is tip to the drop, and now the blood is going to move up in your RBC pipette, and you should have to move up your blood up to the mark of zero point 0.5. Wipe off any blood adherence to its outer side, and you must have to carefully observe that in the RBC there is no the formation of air bubbles in it. Okay, so if the formation of air bubbles, then you cannot perform your experiment correctly. So you should be very careful for about it. Now don't allow blood clot in the pipette. Aspirin diluting human solution is now you are adding this diluting solution whose composition we have seen in the last page. So you have to add it up to the mark of 101 mark now, and make the solution of 1 ratio 200 dilution of blood. Now hold the pipette horizontally. You have to hold this pipette in that position. Now blow out the quarter of the content to remove the pure diluting fluid. So you have to blow out at the end of, for example, consider this is the RBC pipette. At the end of RBC pipette, there is a blower. So from the blower, you blow out the hair, and what you do, because in that side some kind of pure diluting fluid is present. So you must have to blow it out, extract it out. So you blow it out. Prepare the counting chamber and cover it by the cover slip. So now you have to place the drop on this cover slip, sorry, slide, and then you have add up the cover slip on it. Now you place in the Neubaer chamber under the microscope, and now the settling of the cells. The Neubaer hemocytometer scan the counting of the checks always with the 10x objective lens. Then after it, you will use 40x, and you will 100 use, and then you have to count up the number of cells, and count the cells in the 5 medium squares of the small 16 square, one at the corner of each and one in the center. So one in the center is going to represent your RBC. So now the principle of your experiment is given that the RBC cell count, it is the number of RBCs per unit volume of the whole blood. Now the normal values are RBCs are given for the newborn. It should be 4.8 to 7.2 millions. For the adult male, it should be 4.9 to 5.5 millions. For females, it is slightly less, which is about 4.4 to 5.0 millions. For in the pregnancy state, slightly lower than the normal adult values. Okay, so females have more lower values in the pregnancy state, and in the children, its value is 3.8 to 5.5 millions. Okay, so these are the normal values. Okay, so if a patient have changes from these normal values, then you will consider that these patients have some kind of disease related to it. Now the medical concentration, first is the physiological condition. Polycythemia, it is the abnormal increase in the red blood cells. Okay, it should be primary polycythemia vera and secondary polycythemia vera can be occur into the response of hypoxia. Hypoxia means a low oxygen concentration. Last is the anemia. Anemia is the decrease in the RBCs and decrease in the hemoglobin. So it could be due to the lack of production of RBCs, and it could be due to the hemorrhage and some kind of injury. So this was all our practical. Now I discuss the pencil work with you. So in the calculation, you see the count the number of cells in 80 small square located in the 5 middle size square. So you have to count the 5 middle squares, and you have to count 80 small squares into it. The size of 80 small squares is the n. So for the 80 small square, 1 by 20, 1 by 20, and 1 by 10 multiply by 180, so it could be 150, where 1 hour 20 meter cube is the slide line of the square. Okay, so two slide lines, and one is the 1 hour 10. So where the 1 hour 10 is the depth of counting chamber between the cover slip and the ruling. So we have discussed that the counting chamber and the cover slip is above it, so it could have depth of it. So these are the slide lines, and this is the depth, and 80 is the 80 small scale which is present inside the 5 middle size scale. So by multiplying all of these terms we have come out 1 hour 50 meter. So the total number of cell in 1 meter cube is 10 n multiplied by 50 after diluting solution. So the total number of cells before and after should be n multiplied by 5 multiplied by 200. So it could, after multiplying it could be 5.2 million cells. So this is the structure of RBCs, and this is the structure how you will fill your diluting heme solution into it, and you fill it up to the 0.5, and then you will fill the heme solution and fill the heme solution up to the mark 101. So 10 mark is mentioned here. Okay, so in this diagram, this is indicated that this is the middle box, and it contain further 80 box into it. And now you have to add up, calculate all the number of RBCs present inside it. Okay, so I hope that you will understand my lecture. If you want to understand this diagram, then you can visit my previous lecture, and which is about Neubaer human cytometer. So take care of yourself, and मिलेंगे next lecture में. तब तक के लिए अल्लाह हाफिज़.